for joining. Okay, thank you very much for joining the webinar. Uh, I am Wladek Minor, professor at University of Virginia, one of the, together with Zbyszek Otwinowski, we wrote HKL-2000 and HKL-3000 programs. The title of that webinar is the future of structural biology. And we do believe that HKL will have a big role in the future of structural biology. And I will try to show you how to run the programs and how to get maximum out of the programs. There is one thing which is very important, that these days people, the major bottleneck in protein crystallography is protein production. Smaller bottleneck is crystallization, and structure solution or the structure determination, refinement, is relatively easy. And afterward, we have the structure analysis. We have to convert our data into information or into knowledge. Despite of the fact that structure determination became relatively easier, people still have tremendous problems. And if you will look here, and this is something which you have on the screen, is the picture from our paper in Expert Opinion in Drug Discovery about productivity of the synchrotron beamlines in 2011 and 2013. And on the top part of the figure, you have structures, you have all data, and on the lower part, you have SAT, structures solved by SAT or MAT techniques. And you may see something very interesting, that if you will look at the detectors, on the fact whether the beamline is the insertion device or bending magnet, or any how old is the beamline, you see that there is no correlation between any of these factors and productivity of the beamline measured by the number of the PDB deposits. So if you look at all data, the most productive beamline is NSLS X29 in National Synchrotron Light Source in Brookhaven. And this source is going to be closed as a completely obsolete. The next one for all data is APS-19ID at Argonne, which is still 15 years old synchrotron. And none of these have really modern detector. It's ADSC Q315. If you will go to SAT-MAT structure, you have similar situation. You have APS as the most productive. The next is NSLS X29A. Afterward, you have SSRL with Pilatus 6M detector. And so there is really no correlation between hardware which you have on the beamline and the productivity of the beamline. And I do not want to talk about software, but I can tell you one thing, that protocols which are used on the beamline are very, very important. And the most important is how you will modify the protocols to make your experiment. And I will show you the example of the data which I got from somebody, and the structure couldn't be solved for some time. And we will solve that in the next 20 minutes because we have 20 minutes time. And you may have various problems, and you have to learn how to solve these problems. So I will start HKL, and what you have the main window. I will kill that picture. And what you have here is the main window, and it's the same in case of HKL-3000 and HKL-2000. And you have a list of sites. Sites is description of your experiment, and I am checking Pilatus 2M on Electra. There are many gossips that HKL does not work well with Pilatus. I can assure you that it works very, very well under condition that the experiment which you are doing is reasonable. And unreasonable experiment will not work with any detector. So what we are doing, we have main window. Here is the information, where are the data, and we have to put output of the processing into, let's say, sbgrid directory, and we are loading the data set. Yes, that we loaded the data set, and some people are saying that you do not need to look at the diffraction images, that you can just solve structure automatically. And this was probably one of the reasons why the structure couldn't be solved. We display the image because I still do believe that we should display the image. And as you see, the image is not ideal. The crystal is not ideal. And do not read methodology papers, or do not believe too much into methodology papers which are based only on crystals which diffract to 1 angstrom, and or the lysozyme crystals which are close to ideal. In reality, the crystals which you are using have mosaicity 1, sometimes 2, sometimes even 3 degrees, and still it can be solved. So as you see that the image is not ideal, but it's okay. And we are doing first thing, what we are doing peak search. Yes, and you have a number of peaks which were done by peak search, and we will try to index this. Yes, and we are indexing. We are getting some cells. We apply and close. We refine. And we have more or less disaster. Yes? That it's clearly the indexing is wrong. Yes, we can fit all, we can. Yes, and if you are looking what is going on, you see that when you are trying to refine the parameters, detector's parameters, there is something wrong. Yes, because mosaicity is growing up. Yes, and if you will click, you see that only part of the diffraction spots are covered by predictions. So something is wrong. Yes, so there are two approaches. Is to go to another program and try another program, and many people are doing that. The other approach is to think what has happened. And what we should do, we should look what is in the header, what informations are in the header. And what you see here in the header, that 2 theta is 45 degrees. And if you look at the image, it's clear that 2 theta should be 0. So what we have to do, we have override the information which is in the header and put 2 theta 0. Yes? So we override that. We are going to index again. We abort refinement. And we have to do indexing again. Yes, again, we got something. We are trying to fit everything. And we have perfect match of the predicted reflections with the diffraction pattern. This match is not completely perfect. And you may see that when you will look at the zoom window somewhere here. Yes? That this match is not perfect. Why is not perfect? For a very simple reason, that the crystal is not perfect. But again, we cannot expect that the crystal is perfect. The crystallography became more difficult, and it's mainly because it's more difficult to grow high quality samples. And this is something which we, in my lab, encounter every day. And we, as you may look into PDB, and if you will type my name, you will find that we have, during last, let's say, 5 years, we have solved over 500 structures in my lab. In my lab alone. I'm not talking about structures solved in the center, one of the centers. It's in my lab. So we have substantial experience, and as I said, the number of very good crystals is very small. So we are doing few refinements, and you see that mosaicity is reasonable, and mosaicity is not growing. Yes, and the match between predicted reflections and diffraction pattern is very, very good. And also you may see that we have diffraction more or less to the edge of the detector. So we can check various things, but usually program works quite good on defaults. We can check whether the integration box is okay, and it's more or less okay. It could be little better, yes, it could be little better, but it's more or less okay. Yes, we could increase spot size to 40, and probably it will be little better, but this is not critical. Now we have to check whether we do not have higher symmetry, and it's probably primitive orthorhombic. We are not 100% sure of the space group. So we are processing P222, but we have only geometrical information now. We do not have intensity information. So we have information about Brave lattice, not about the space group. And we will start integration. And if you click integrate, it moves you to the next window, which have the information about the integration. And you can follow parameters. I mean, you can follow x and y chi-square. And as you see that the crystal is not ideal. Yes, so your chi-squares are not equal 1. They are moving because you have more than one crystal in the beam, and these other crystals affect your your data. And again, you could grow better crystal, but we will see if you can solve the structure with the crystal which we have. If you here click some other parameters, for instance mosaicity, you see that mosaicity is anisotropic. Yes, and it's again, it's not pathology, or one can say that it is pathology. It is something which these days is normal, because all projects which were easy and which were giving perfect crystals were all already solved. So you cannot work on that. You can, but it does not have too much sense. Or they are not interesting. Yes, that people didn't do that because they didn't think that this protein has any biological relevance. So again, it may happen, that your crystal, that you can grow perfect crystal from something which is biologically important, but it does not happen too often. Usually, I would say that this is very good crystal, despite of all these problems. This is very good crystals, and despite of the mosaicity. So we process the data, and we will solve structure by SAT. And we are doing scaling. And we are scaling 360 frames here. And I'm not going to show that here, or because we have only half an hour, but if you would have 180, you would solve these structures equally well. So now you have very small fraction of reflections which are marked for rejection. And you see, you clearly do not have radiation damage. So we will put fix B, and we apply small absorption correction, and we will scale again. And here you can see completeness versus resolution, and you see that the data are complete. The only problem is that when you are going to the higher resolution, the data are weak. So this is 3I over sigma. So data are nominally to 1.5, but only to 50%, you have only to 1.8. I mean 50% completeness of reflections which are higher than 3 in I over sigma. And here you may look at your chi-square and R merge versus resolution for merged Friedel pairs and unmerged. And if you have this difference, it indicates that you have anomalous signal. Yes, that you have anomalous signal. And you really should look at chi-square, not into R factor. And here you have some other statistics, and this is chi-square and R merge versus average intensity, average cumulative redundancy versus resolution, I over sigma, and the CC half, this new Karplus statistics. And okay, and now we will say that that we will apply autocorrection. It's a new correction for radiation decay from various pathologies which you may have in the detector, not necessarily pathologies, and you will use this reflection. Will scale again, and in the last scaling, we will reject this very small number of reflections which do not fit. One thing which is very, very important, you have to, if this number is high, it's red. And if you have red, it's just the warning that if you should have more or less 0.1% reflections which are marked for rejection. And you see that your crystal is not ideal. If you have high number, you should think why. Yes, and there may be various reasons for that. But now, after correction, you have only 77 reflections out of 500,000. You have only 77 reflections marked for rejections, which is 0.02%.
Vlad, can I interrupt for one second? We had a question while we're on the subject here. I think maybe this is appropriate. Someone asked why you fixed the B.
Because you see, I didn't see the B factor was going up and down within point half of the angstrom square. So it was clear that you do not have radiation damage. If we would have the radiation damage, I wouldn't fix. If B would go up, yes, we would have radiation damage. There is no point to add parameter when this parameter really artificially change. Yes, you did I answer correct? I mean, I know that I answered correctly. Did I answer the question for the?
I think that's a satisfactory answer.
Okay, great. So now we will do that again, but we do rejections in the last scaling. And we are removing 77 reflections only. But we should remove them. We should remove them because if they are at low resolution, they may affect phasing. So basically, now this is the last, almost the last scaling, but you see that already, and this chi-square should, that already you have tremendous difference between merged and unmerged chi-squares. So we have very strong anomalous signal. Now the question is, what is the space group? We have P222, but it may be P21, 212, it may be anything. Yes? The best way to find that is to look at the reciprocal lattice, and you see that this is L and H, and you have every second reflection missed on L and H. Yes, so now let's do on H and K, and you have the same situation. So it's clear that the space group, or it's very, very suspicious. The space group is P21, 21, 21. And we will scale again in that space group, and this will be the last scale. And now we are scaling. Obviously, the scale will not, it will not change anything. We are just removing some reflections. But the result of scaling, the statistic from scaling for P222 and P21, 21, 21 will be exactly the same. So you cannot make a distinction on scaling. You can only make distinction by looking at your reflections among I'm sorry, along H and K axis. So okay, so it's done. So the next thing, we should go to the structure. But if we would like to go to the structure, we have to tell program something about the project. And in fact, we should have done that before. We should before enter the sequence, and this is the sequence. And and we should tell one more thing, that this is what technique we will use to solve the structure. It may be SAT-MAT, molecular replacement on ligand screen, or we should say something if this is protein, small molecule, and virus. And because of lack of the time, I am showing here only for protein, and this is selenomet. So program figures out that we have 7 selenometionine, because we have, in fact, we have 8 here, but first methionine is at the beginning of the, so it's probably very floppy. So the program assumed that it's 7 in first attempt. And we are going to structure, and in structure we will click data analysis. And we see that we have, after averaging, we have very high anomalous signal, because this is the noise level. And we have good completeness. We may look at some other statistics. We can check twinning. And the next thing, we will try to find sites. And we are using ShellX to do that. And you see that the program found the same site 15 times, the same very high correlation coefficient, and the same position 15 times. And at that point, program just stopped. Yes, because there is no point to continue that. And we may look at sites, and these are the sites, or we can look at the graph, and we have really not 7, we have 8 sites. But 8 sites is very weak, and this is the site. I know that this is the site on the, and we could kick that out by clicking, but we not necessarily will do that. And we are going to phasing, and the first thing in phasing, we are checking the, in jargon, we are checking the hand of the solution, or we are checking which enantiomer is correct. And here we have a map contrast. We are doing solvent, we are checking that by solvent flattening. And you see that the map contrast for one enantiomer is much higher than the map contrast for another one. And this is the best indication that your solution is correct, that your sites are correct. I mean, it's still an indication. Check here that everything was figured out, yes, from the structure. So it assumed that it's one molecule in asymmetric unit, that solvent content is 0.38. We can change that. Yes, we can go to 2. But we are getting unrealistic, yes, completely unrealistic solvent content. Yes. And now, when we found that we should phase the program. And first, what we are doing, we are repeating solvent flattening, little better than before. And this initial solvent flattening is done by ShellX. And we are using various programs, and you, to give the credit to all people who wrote these fantastic programs, here you have the information what program is run. And to phase, we are running MLFair, and you can look at figure of merit versus resolution, or on phasing power in every cycle of the refinement. And here you have digital information about the refinement of the sites. This refinement of the sites is quite sophisticated because some sites, some weak sites are rejected. So we took 8 sites, but in fact the 8th site was rejected during the refinement. Yes, it was judged that this is weak site, and we will rather lose than gain. I cannot talk. Could you call in 15 minutes? So you see that it's we are still doing refinement, and now we are doing solvent flattening, and solvent flattening is done in that case. This very accurate solvent flattening is done by DM. I'm sorry, I didn't. And now you have a map. Yes, you automatically map is displayed. And you see that this is beautiful map. Yes. So this is the map. What you have here is the anomalous sites, and these anomalous sites are very reasonable. Yes, and you have holes in aromatic ring. Take into account that this is experimental map. This is experimental map, not the map after refinement. This is experimental map. So the only thing what we can do is to build the model, and again, we are doing click. Yes, and we have to wait a moment for model to be built. So what is the best check if your processing is correct, if your data reduction is correct? Is to solve the structure. And you see that you may get fantastic R factors from the processing or from the data reduction. But if you cannot solve structure, something is wrong. So you see that, remember about one thing, that your goal is not to collect data. Your goal is not to process data. Your goal is to solve and refine structure. This is your final goal. And this is just the example of one of the structures which I got, not my structure, but structure which I got from somebody because he couldn't solve that structure. And as you see, this could be done. So now we are using ARP-warp to build the model, and you see that in the first cycle, 80% of the structure has been built. Yes, so it's clear that we will have structure very, very soon, and we will be able to refine structure very, very fast, and we will be able to deposit structure very, very fast if our goal is deposition. But our goal is usually not just the deposition, but our goal is to analyze the structure. Yes, is to analyze the structure. And we have to, and this may require a lot of effort. You may use different techniques as SAX or electron microscopy or ITC or kinetic experiments. And you see that to solve the structure is not enough now. Yes, you have to analyze the structure in the context of the function. And if you will look at our papers, quite often we have we are using many techniques, not only crystallography, to get to gain some biological knowledge. And you see that here, what we have, as I told you, 80% of the model is built, and now you have refinement, and R3 is 30%, R is 27% after first cycle of the model building. And as you see, the R and R3 is dropping very fast. Yes, because R3 is already 26. Yes, and there will be few cycles. I do believe that I said, I run everything on default, and as you see that R and R3 are dropping very fast. And in the next cycle, we will have much better model and much better R and R3. So even without touching the refinement, even during the model building, we are very close to the results, to R and R3 results, which many people saying that they are final. And again, your goal is not 20% R3 for this structure. Your goal is around 16% R3. Yes, this is what you should get from this. And we are starting building model 2, but I already will show you oh, I'm sorry, I made mistake. So you see that this is the model after first cycle. Yes, after first cycle, this is the model. Yes, and you have everything almost completed. And you see that very nicely the anomalous peak in the map, on anomalous map, is in the place where is methionine. So this is additional way to check that everything is correct. And we are looking at that, and we have a model building again, and hopefully structure won't be completely done, so you will be able to do something by hand. Because remember that you will be the first author of the paper, not the software. So software shouldn't be completely automatic. And you see, we could do that automatic, and I could run you that on automatic mode, and you may look here. You have click auto. You can click auto. You have auto mode. But we do not distribute auto mode for a very simple reason, that if you can solve the structure in 20 minutes, and you are not doing that too often, yes, you are not doing that too often, there is no point to do that automatically. You are doing that semi-automatically. And the program stops in 6 places. So it's some people call that 6-click model. Why? For a very simple reason, that you should try to engage your brain. You should look at the results and try to solve the problems if there are problems. Yes, if there are problems, we can go back to structure, and you see that it's again uh, now 83% of the model is built, and uh, and R3 again dropped substantially. And after model is built, you may go to refinement, and you can look at geometry, you can refine the structure very, very fast. So the process of from the moment when you have a crystal in the beam to the moment when you have refined the structure, refine and validate the structure, can be very, very fast. We have 45 minutes. I'm sorry I extended that. If there are any questions, I would be happy to answer the questions.
Great. If anyone has questions, feel free to send them in the chat window, or we can answer them. Thank you very much, Wladek. I had a couple of questions myself. One from earlier in the talk, I know with recognizing anomalous signal, when you're on the beam line and you've got your crystals up there and things are often radiation sensitive and the resolution's modest, or maybe you have some derivative that's partially occupied. You said chi-squareds can be better than R merges for seeing anomalous signal. Can you talk about that a little bit?
Yes, because you see, R merge is unweighted statistics. Yes? R merge is unweighted statistics, and if you are looking here at scaling, the R merge, there is some difference between Friedel pairs, but the difference is very small. Because you are not taking into account the error. Yes? If you calculate chi-square, you are taking into account what is the error. Yes? What is the sigma of the intensity. And for that reason, you see very, very clearly that you have strong anomalous signal. Yes, so you see that you said that in reality you may have a problem at the beamline, more problem than here. Not, but I disagree. That this is the norm. I mean, this diffracts much better, yes, because it diffracts in the end to 1.5 angstrom. But we are not taking crystal out of the beamline until we have 50% of the model built, at least 50%. Yes?
Right.
Or the other possibility is that the crystal is completely dead.
Mm-hmm.
But we are trying to really not to get images or not to get even scale data, but we would like to have partial model. And not always it works. Yeah? So we had, there is a few years ago, we have a paper which had in title Crazy Math. And this crazy math was done on that way that the first day of the data collection, we collect the data, we built 50% of the model, we took the crystal, and we found in the next 24 hours that we cannot complete the model. So what we did, we put another crystal on different beamline with little different wavelength, and we use these two data sets as a math. So you see that something which is crazy according to the standards which were used a few years ago. And the structure was solved without any problems. We have 90% of the model built after the additional data set. Probably, if we would merge this data as Wayne Hendrickson is doing now, we also would solve the structure. Yes, so you see that sometimes you may not have enough data. Sometimes you would like to merge two crystals or more crystals to solve the structure. But sometimes we have just the one crystal. Yes, we have just the one crystal. We are not able to grow more than one crystal. Okay, and now this was finished. This is result of the 3 cycles, and this is the result. And you can go to refinement and try to refine the model. And it's again we are using RefMac, and we may check geometry. We are using mole probability for that. We are you really may have great help in the refinement. And here, if you look, these are your goals in the refinement. So we have already 17%, little over 17% are, and 20% are free. Yes, so we are very close to structure completion. Yes, I mean some of my people can do that in few hours from that point. Did I answer your question?
Yeah, yes, you did. And we have one more question that came in. I think this is referring to the indexing process when you're maybe in a tough indexing, sort of non-ideal or crystal. When you've got red rejected spots that are flagged as rejected, what's the best way to open up the parameters to sort of help increase the number of acceptable spots?
Okay, I mean, first of all, you should have all spot. I mean, the red spot should be only if you clearly see overlap between two spots or you have faulty region of the detector. So in that case, if you will look at the, do I have that still? In that case, no, I killed I'm sorry, I killed that. What you should have very few red spots. I mean, in case of Pilatus, you may have red spots between panels. Yes, or for some detectors, people are trying to hide the place between panels, and you may have some red spots. If you have red spots during indexing, in the place where you shouldn't have red spot, it means that something is wrong. And you see, I think that I shown here enough, so I will go back to illustrate that. Yes, and we may just do refine. Yes, it remember about indexing. Yes, and this is the result of the refinement. So you see that you have red spots here. Yes, and you should have red spots. Yes, because there is no diffraction image here. But still there is a space where the spots are predicted. And because not all Pilatus are the same, we cannot mask these spots. Yes, on any other way. And you have here one red spot, and this is the reasonable red spot, yes, because you have the difference between beam stop. Yes, this is partly the beam stop, partly. So you see the program decide that the variation of the background is so high that this spot should be rejected during, and the same is here. So basically, you do not have red spot. Now, let's do something artificial, yes? Why? We had reasonable side configuration. But let's put here, instead of beam position 145, let's put 141, yes? And let's try to index. Yes, you still have some indexing, and you may apply and try to refine. And you do not have here many red spots still, but you see clearly that if you do profile fitting radius and you observe only reflections which are higher than 5 of I over sigma, you have many reflections which are not predicted. Yes, so something is wrong. And again, if you will look here in zoom window, yes, it's a good possibility that your position of the beam is wrong. That position of the beam should be somewhere here. Yes, and somewhere here is 145.8, 126.7. So when 145 instead of 141. It does not need to be completely accurate. Yeah? So we corrected that. We abort refinement. We are doing indexing again. And now it's perfect, close to perfect. Yes? And this is refined position. So it's not exactly like the positions which we put, but very, very close. So if your position of the beam is not better than the half of the distance between spots, if the error is larger than the half of the distance, you will have misindexing and you may have red reflections. So we can make position of the beam completely wrong, 130, and abort, and try to index. Yes, and you have something completely wrong. Yes, if you will refine long enough, you will have red spots. Yes, because it cannot refine and the mosaicity may grow or something else may happen. Did I answer satisfactory? Hello?
Sorry, I was muted. They say yes. Thank you for answering. And I think with that, if there aren't any questions, I think, thank you very much. That was very useful, very informative. Good to see the data set to model in 30 minutes and 6 clicks. Yeah, very nice.
And structure.
Yeah, exactly.
Okay. Okay.
Right.
Thank you
Great.
very much.
Thank you very much.
Thank
Thank
you.
you for joining us.
Bye
Bye.
bye. Hi, Jason. Okay, how many people did we have?
Your audio is still broadcasting.